Hey guys, Dr. McDonald again, and we're going to do Lab 10. This part of Lab 10 is going to be about how to do a series of biochemical tests on gram-negative bacteria. So, if I were given an unknown bacteria, first thing I would have to make sure is that I've got a pure culture. I'd have to make sure it was isolated. The next thing I would have to do would be to gram stain it to characterize what it is. So say I've done that and now I've got a gram negative. How do I tell what which gram negative this is? Well, the only way I can tell the difference between, say, E. coli and Pseudomonas aeruginosa is if I check them out with biochemical tests. Now, we've got several that we're going to be talking about in here. I'm not going to go over what's up here in this introduction because I'd rather go into it when I actually talk about it. But basically, we take these guys and we run them through these tests. And these tests will give us results which will either tell us exactly which bacteria we have, or they'll tell us what is the next test to do in order to continue to narrow down. So these are for the unknown bacteria cultures. Now, you guys aren't going to be, you guys will not be using this like we do normally in the actual lab. And I'm thinking I need to check on it because I'm about to give you guys the unknown lecture or the unknown lab information in just very short, probably by the end of this week. But this is where we're going to be with this guy. We keep narrowing it down. Now we have, they call these guys working dichotomous keys. What does that mean? It's a big long word. It's basically a list that tells you the test results for all the bacteria by species. So if you get a certain result, you can either say that's exactly which bacteria, or you can say, well, now we need to do this other test. So we'll talk all about these as we go through them, and I can give you some insight on some of these. Now, let me also say there's a lot of links at the bottom of this lab for all these different tests. I realize it, but maybe what you don't realize that a lot of those are like 1 to 4 minutes. They're very, very short. So I would definitely go through and watch those. It'll be worth your while, guys. So we've been using the TSA, the tryptase soy agar, or that nutrient broth that in the test tubes. Those are for the more common type of bacteria, but there are certain media which we call complex media, and these guys are going to have special nutrients or other things that these special bacteria need to grow. Example is our SIM agar, EMB plates, so forth, so forth. Complex media help us identify bacteria by selectively allowing only certain bacteria to grow on them. The EMB plate, oh well, that's in the next one. I'll go ahead and defer until we get to that. But anyway, complex media have special nutrients or growth factors that specific bacteria need to thrive. Now the selective media, and yes, the media can be selective and complex, they have certain substances which will keep some bacteria from growing. Eosin methylene blue, EMB agar, these two dyes, eosin and methylene blue, are toxic for gram positive. So that means on an EMB plate, you will be growing a gram negative. We don't use this to classify. We use our gram stain. but still, it's a good thing to have. And besides all of that, not only is it selective, it's also differential. So if the gram-negative bacteria happen to be the coliforms, the the gram-negatives that live in the human colon, these guys have the ability to break down milk sugar called lactose, and this makes them different from a lot of the other gram-negatives. So EMB plates are also have lactose in there. So when these guys, the coliforms, Enterobacter aeruginosa, E. coli, when these guys take that lactose and break it down and use it for food, and they produce a waste product, this is going to cause these colonies of these types of gram negatives to have, they call it nucleated. It's got a little center section that's going to be purple, dark purple. Could be pink if it doesn't if it breaks down lactose but not in any significant amounts. All the other bacteria, gram negatives, are going to be clear that don't use lactose. So this kind of helps us, the dark purple centers. E. coli, in addition, E. coli, because of the acids it produces, has a beautiful emerald green sheen that looks, I mean, it looks metallic on the plate. Uh, yeah, pink, maybe not non-fermenters. There'll be less fermentation. So by using EMB, we can help to classify some of our bacteria. We can break them down into lactose fermenters and non-lactose fermenters already, in addition to weeding out any gram-positives we may have. Okay, now metabolism, and this is going back to these. We also have tubes which contain sugar. Got some that have glucose, some that have lactose, some that have sucrose, all different kinds of sugars. We usually use about 4 different kinds in the actual lab. And so you can take, that's a liquid culture with the sugar in there. You take bacteria and you inoculate it in there. These guys are red when you get them. And if the bacteria can use that sugar, they will produce acid, which will turn it yellow. And so by turning it yellow, it lets you know that that bacteria can ferment, can eat that sugar. And again, that's really important. In addition to which, there's a little tube in there that is upside down, like an upside down tube. And when you get it, there will be nothing there. There'll be fluid all the way to the top. But if the bacteria, in addition to using the sugar, if it produces gas, usually you'll see a bubble inside that tube. That's called a Durham tube. We also have gram-negative rods if they're part of the Enterobacteriaceae family. It's just a genus of bacteria. We also have something called the Enterotube 2. It's actually called the Enteropleury now. And this is a long plastic tube that actually has 12 sections of agar in it. And you actually have a long rod that goes through the middle of it. You get the bacteria on the end of the rod, and then you pull it through the tube and push it back through. And by doing that, you inoculate these guys and all of those. And between those 12, you get 15 different biochemical tests. Now, this guy comes with a little booklet that gives the interpretation. And this is a one-stop thing, guys. If you got reason to believe you have Enterobacteriaceae, you do this and you're done. So here's some of the bacteria that we normally give out for the gram negatives. The lactase, sucrose, glucose, fermentation tubes, maltose, that's the other sugar. MRBP tube, let me come back up here. The Voges-Proskauer test for acetoin, and the methyl red test for acid fermentation. So what you do is you actually inoculate the bacteria in the BP tube. You let it grow 48 hours. At the end of 48 hours, you bring the tube out. You take a pipette and you take drops from the BP tube, put it in a clean test tube, and then you put the methylene red in there. And again, if it turns red, it's positive. And here's something that you might want to remember. If it's positive on methylene red, it's going to be negative for Voges Proskauer. You'll also, after you have removed the stuff for the sample for the MR test, then you've got two reagents A and B, that you put drops in there and let them sit for 15 minutes and see if you get a positive for this guy. Again, positive here means that they produce acetoin and they did not produce acid. The SIM tube is a test tube with a slant that you inoculate. I think I really will talk about this on these guys because they're pretty complicated. The EMB plate and the enteropleury is what it should be saying. Okay. Naturally, when you get your test tube, you put your initials, the date, and the suspected name of the organism. Basically, I only tell them to put unknown 1 or unknown 2, whichever one you decided was gram negative. You inoculate all of these guys, and again, it's just, and you really don't have to do all 4 of these guys. This is for the unknown. For this lab, yeah, you do all 4 just so you can see the reaction. But when you're actually doing an unknown, you've got that dichotomous key. There's a flow chart that shows you which test to do next. So not every bacteria needs a lactose fermentation. But in any case, the MRBP also, there are indications for using that. That's acetoin is a precursor for this. The SIM is one of the ones we don't use a loop. We actually use a needle. We stab it straight down. We're looking for 3 things. We're looking for sulfur, which is going to be in the form of black granules or even the entire or blackness in the agar itself. We're looking for motility. That's what the M is standing for. So when you put this down, and after it's had a chance to grow, you look to see if it has spread significantly beyond where that needle thrust was. And in the indole test, you actually put some drops of reagent and you look to see if there's a reaction. So there's a SIM test. Now here's something else. On the EMB, we don't do the streak plate to 3. We just do one single line down the center. Enteropleury, guys, I'm going to be honest with you. I think you really need to watch the video on this because I've already explained it, but it's really something that you could better see and maybe understand by watching someone do it. We also break part of that wire that's in the middle the handle part. We use it to punch holes. There's some thin plastic covering on 8 of those compartments. We need to punch holes to allow oxygen to get in. And basically, you put the entire enteropleury in the incubator just like you would the petri dishes. Okay, so the fermentation tubes, that's really easy. It's yellow or it's not yellow. If it's not yellow, it's negative. Gas, it's either positive or negative, real easy. Let's see. Now, for the MRBP, this is going to be a little bit different. And these are actually going to start off as yellow. And you're going to take the drops out and put it in the clean test tube. But it'll actually turn red if the pH is lower for that methyl test where you drop the methyl red in there. This, guys, need to keep these guys from being confused. The sugar tubes, fermentation tubes, start off red, turn yellow positive. These guys are going to be yellow and they turn red for the positive. And again, if you got a positive on the MR, you should have a negative on the BP. And butanediol is the product, and again, we're checking for acetone. Reagent A is going to be the naphthol. It produces a pink burgundy that you're going to see in there. Oh, by the way, let me make sure I tell you, if it turns out, if it doesn't change from yellow or if it even changes to orange, it's still negative. It's going to be a deep red if it's positive. Anyway, naphthol is we're going to get this pink burgundy color, and that'll give us a positive on the BP. The alpha naphthol is the reagent A, reagent B is potassium hydroxide. This is a long test. In the lab, I tell everybody to do this one first if that's one of the tests you need to do, because it's going to take 30 minutes possibly to get a reading. And of course, the reason we don't put these in ahead of time while they're growing is because they're toxic. So in the BP test, now let's do the MR test. We placed 16 drops of that MRBP in the clean test tube, and then we add 5 drops of the methyl red. We don't mix. Red color at the top, positive mixed acid, no color change negative, or even an orange is negative. Now, after we've done that, and we've got to take this out before we put the BP reagents in. That makes sense, I hope. You put 8 drops of the naphthol and 4 drops of the KOH. Mix gently, and again, 20 to 30 minutes is more accurate. Okay, SimAuger, sulfur, indol, and motility. It turns it black, and it can be even granules. It's sulfur positive. Indol, we put something in there. It's called Kovacs reagent. And for motility, again, we just look to see these guys that are moving away, and it may be even cloudy. In some cases, the whole tube will be cloudy, but non-motile, they're just going to be in the line of the where the needle stabbed down. Okay, after you got your sulfur and your motility results, then you add the 5 to 10 drops of Kovacs. Shake gently, and again, red is positive. EMB, selective and differential, we talked about this already. Eosin, we talked about that. Fermentation by lactose by coliforms, dark purple centered E. coli has the metallic green. And here we don't have a video there for you guys on how to interpret this. Again, it's in the book. You go by compartments. First compartment is glucose fermentation, just the same as the tube. Yellow is positive, red or orange is negative. Again, yellow and red, sometimes whether it's positive or negative depends on the test, so don't forget that. Interpret the results of gas. If the wax on the agar has been lifted up away from the agar, then it's positive. If it's not lifted, if it's still in contact, it's negative. And sometimes it's very subtle. Compartment 2, lysine decarboxylase. Any purple is positive, yellow is negative. 3, interpret the results of ornithine decarboxylase. Purple positive, yellow negative. Here's our hydrogen sulfide production again. True black is positive, beige is negative. And also the indole production. We make a hole, and we put some Kovacs just like we did on the Sims tube. And if red pink, it's positive, no color is negative. Compartments 5 to 8, adonitol fermentation, lactose, arabinose, and sorbitol fermentation. Any yellow is positive, red, orange, negative. And we've even got a BP test here, unless we don't really use this unless we are calling for a confirmation test. Compartment 10, dulcitol fermentation, yellow positive and yellow color negative. Phenylalanine deaminase, black or smoky gray positive and yellow color negative. Compartment 11, we're going to interpret the results of urea hydrolysis. Red or purple is positive, beige is negative. If it's able to take urea and break it down, this is something that you find in those bacteria that cause urinary tract infections, by the way. Compartment 12, citrate utilization. Blue is positive, green is negative. So we circle all these. We add and there's circles on the actual results. There's a form that goes with it. You add those up together and you take those 5 digits and in the identification book, and it gives you the information. Okay guys, I've been to all of these sites and these are very short for the most part. So do go and view those, because I realized that despite my best efforts to verbally describe this, that a lot of these are things you need to see. Alright guys, that's got it for this one.